Okay. Welcome everybody. It's exciting to be here for another VICC General Call and see many familiar faces and a few faces that are kind of new to this group. Today we're going to have a guest presentation from Andy Yates. Andy is he's the forgive me if I get this wrong, Andy, the Genomic Information Sciences lead at Ensembl-EBI.
Kind of, sort of. So,
And he's going to be talking about the work that he does wearing one of his other hats, which is as the one of the co-leads of the Genomic Knowledge Standards technical workstream within the Global Alliance for Genomics and Health. He's going to be telling us today about GA4GH and about GKS and how it operates within GA4GH. Before we dive into it, though, just a couple of quick announcements. First, the ClinGen Curating the Clinical Genome conference abstracts are due today. So if you have not submitted yet, please submit. And second, the Cancer Genomics Consortium annual meeting abstracts are due, I think, at the end of this month. And of course that's rapidly approaching. It's in like 9 days. So please, if you're planning on going, and I encourage this community to go, think about submitting an abstract by that date. Okay. So with that, I'm going to hand over the floor to Andy. Show is yours.
cool, thank you. Yeah, sorry about the title as well over at EBI. This is what happens when you get to invent your own title. It's never good. Just let me pull up the slides there. I'll do the normal Zoom thing of asking if everyone can see that. Yep, cool, excellent. So yeah, I'm going to have a quick chat today. Hopefully, I did go through the slides and there's probably way too much content here or so. So if I gloss over things, then please pull me up on it if you find it interesting. But I'm trying to give a flavor here of what's going on in the wider Global Alliance and a little bit of context as well. So before we go anywhere, Alex did a good job, a brilliant job of actually introducing me, to be frankly honest. So I am based over in the UK at Ensembl-EBI. I'm co-PI of Ensembl and Decipher. Actually, I'm on the curating the clinical genome organizing committee as well. And actually, my main day-to-day job is part of Ensembl's massive infrastructure refresh that's going on at the moment over there. But with Global Alliance, I've been involved since 2014. So before the massive reboot in 2017, I became a workstream lead alongside with Bob Freemuth. And as part of Global Alliance, I've gone and developed 3 standards as far as I can remember, which is VERST, RefGet, and Service Info, and now provided some additional support to Susan Fairley, who is the current Chief Standards Officer of Global Alliance. So in terms of the organization, now hopefully this does not come to any kind of surprise, is that this kind of model here, the Global Alliance, is formed of the wider community. Everyone here is part of that wider community. There are funders that actually help to support not only the community, but then also actually staff. We have a set of host institutions. We'll go into what those host institutions are, and actually an incorporated elements, which is kind of in all of this as well. But effectively, we are funded in part to provide Global Alliance, but actually Global Alliance is still primarily a volunteer-led organization where all of you are here basically as part of the driver project, donating your time to this effort. So thank you very much for doing that. Host institutes, we have 4, OICR over in Canada, the Broad in Cambridge, USA, and then the 2 Wellcome Sanger and Ensembl-EBI, which are both located on the Genome campus, which I think I know a lot of you here, and you've actually been there as well, so you've already got an idea of all of that. There is also a large component of paid for staff. So these are based at the host institutes I mentioned previously. They roughly move into 3 areas of responsibility through to project management, policy. Sorry, 4. I can't count today. That's great, that is. Communications and operations, and also the technical side of things as well. Beatrice is on the call as part of our project management team as a workstream manager. These are all supported by grants, which come into each of the different host institutes. And then Peter, Angela, and Susan coordinate the effort out of those. Beatrice has got, is that hands up or a wave? Oh, that's a wave, isn't it? Sorry. So anything that you want to ask questions of about Global Alliance, Beatrice is kind of your local person that you can ask those questions. And I swear there are no stupid questions in this game whatsoever. We also have a huge amount of driver projects. We had a number of those join at the beginning of the year as well. Driver projects obviously being the source of drive behind Global Alliance, providing real use cases and context to the standards being developed. And really, driver projects, whenever we go through product approval or anything like that, there is this massive drive in the background to actually say, okay, well, who are your driver projects? Who are you serving? Who's the community that's being served here? So driver projects are incredibly important towards how we actually work in this organization. And there are a few that have come through this time around that I just want to point out as being particularly interesting, I feel, as well. So we have things such as the Human Pangenome Project, which is actually, it's not a spin out of HPRC, but it is very much related to HPRC and efforts around pangenomics in human. And also the Genomic Data Infrastructure, which is an EC project being led out of the Beyond 1 Million Genomes project, which effectively is trying to deal with what happens if you try and get the genome of a ton of people across Europe and beyond. Well, actually, sorry, within Europe, but beyond 1 million of those genomes. Now, there has also been a change of leadership here in 2024. Ewan, who is Director of Ensembl-EBI, steps away from the chair of Global Alliance and has become chair emeritus, with Heidi Ream, who is based over at the Broad and is actually Driver Project Champion of ClinGen, has moved up into that leadership role. This has really given the organization a bit of a refocusing, but ultimately it's still roughly the exact same direction in which it was going in previously. Something else as well that may or may not have come up on people's radars is actually my co-chair, Bob, is stepping down this year. And we're looking towards actually finding a new co-lead for the work stream as well. So this is kind of a little bit of a call of saying that if you're interested in this kind of thing as well, then please reach out. But Bob will be certainly missed, but he's not going away. So what kinds of things have actually been happening in the background? So one of these is actually a gap analysis and strategic refresh. So back in 2020, Heidi was instrumental around actually conducting a gap analysis. So understanding where are the gaps between the Global Alliance standards, our products and external standards, and looking for ways to actually support and improve the implementation of that and to actually align a bit closer with healthcare and clinical standards. There was also a strategic refresh, which is attempting to create this forum for experience sharing and implementing products, and I'll go into what some of those outcomes have been, but then also to actually try to work more with collaborative projects and to ensure clinical relevance of the toolkit. And effectively what this has led into is actually a refresh in how we structure our standards development that was led by Susan Fairley. But ultimately what this means is that we now have structures called study groups. Study groups allow us to have focused discussions around tight subjects with a defined end around that. We have mechanisms to actually create standards and frameworks and policy frameworks, which is always lived in the work streams. But then also we have the GA4GH implementation forum, which is attempting to implement those products to solve those problems. And in the middle of this, we have things like national initiatives and communities of interest, which we'll go on to as well. And I believe that everyone here should at least be aware of one community of interest. So what are the areas that Global Alliance is looking towards over the next 3 years? One of those is actually a focus on implementation and data sharing. So variant level matching, federated nomad data. Hopefully that is everything of interest here, especially variant level matching. Adding new communities of interest, one of those being clinical laboratories. A new one that has popped up is actually how to endorse external standards when they're relevant to Global Alliance, and that is actually going under its first review now internally. So if you know of any standards that you're thinking, hey, yeah, that's a standard that Global Alliance should recognize, or Global Alliance has included in its toolkit. So a good example here would be, say, HGVS. It's not Global Alliance standard, but Global Alliance standards build or use HGVS. We also have an aspect around supporting the wider communities, grant applications and supplements, and also a strategic leadership committee, which is a new group which will basically start to meet with the exec and provide strategic guidance. This is to kind of sit a level beyond steering committee. And we're looking for individuals that have extensive experience of Global Alliance with a forward-looking strategic view. And that is currently open. That call is open at the moment for applications around that. I think the Strategic Leadership Committee is going to be a really, really interesting group in how that actually works and how it interacts as well. There's also a set of driver project workshops. This is just a dump of everything that came out of our last steering committee slide. If there's anything here that you think, yep, you know what, that looks interesting and I don't see ourselves actually on there, then please basically pop your hand up. Otherwise, then you don't have to worry about it too much. I will pop up a couple of these other ones later on though as well, because there's a few that are interesting when we get into a little bit further into that. But GKS is placed within Global Alliance. So we are, as it loads in, one of the technical work streams. There are 8 of us. Well, I should say there's actually 6 of the technical work streams, and there's 2 foundational work streams. Data security and the regulatory and ethics are considered foundational, but then we have these other 6 hanging around, which are, I say, hanging around. That really isn't doing too well for them. That really underpin the other development of standards. In terms of genomic knowledge standards, we are developing downstream, we are enabling that downstream analysis of genomic data, developing common APIs for exchange and reference genomic information. This absolutely fits in exactly what VICC has done, which is why VICC has been a huge driver project of ours and Alex's. And I'm trying to look through other people, Wes as well. Brian, oh my God, so many people who actually have been contributing into that work as well. So VICC very much plugged into genomic knowledge standards. In terms of the items, I said items here. Originally, this was standards, and it kind of goes beyond that. Those areas that we've looked at or are developing at the moment are things such as the variation representation specification, its accompanying specification versatile, variation annotation or variant annotation, which is about linking knowledge to that data, which is being led by Matt Brush and Harvey from Genomics England. Sequence annotation study group and the categorical variation study group as well, which are 2, well, sequence annotation has been going on for quite a bit, but it's trying to break down the problem of how do you consistently share genomic annotation across multiple providers without going crazy and without having to use GFF. And categorical variation is attempting to basically pull into an area that I know has been of great interest to Alex. I'm being quite vague about that because actually we'll go in a bit further for a bit more information on that. Now, the next few slides, if you're at the last steering committee meeting, just go read a book for a second or so, because this is going to be a repeat of that information. But the areas that are currently being looked at, and this is actually moving towards one of the working meetings in April, which is called Connect. We have 2 Connects each year, which is meant to be dedicated to face-to-face time for actually working more on these issues within Global Alliance, and one of the ways that Global Alliance actually facilitates work outside of these meetings that we have here. One of the things that Alex and Larry have been very passionate about is the idea of this maturity model on the right-hand side, about how on earth can we actually start to describe standards in a manner that actually makes it clear on how well implemented they are, how well adopted they are, how stable they are, and effectively how can we encourage a level of confidence in what's being developed there, and how can we ensure that adopters and developers basically when they come into something they understand the stability that accompanies that. So this is a massive thing and it's sorely needed for us. It's still under quite a bit of discussion as well, I know that much, but it's getting there. We also have, we are also moving towards our version 2, which I think is best described as what verse 1 did. Let's see if we can do it slightly easier in verse 2, but then this has also gone a little bit further as well to start to develop some more structural variation foundational classes for verse 2. And also there is a certain amount that basically is coming from CatBar as well. But if you come into Connect, there are some good sessions coming up there as well, which I don't know, Alex, if you're going to mention about Connect or anything, but I'll just leave it there for the moment. Categorical variation study group is really about trying to classify effectively the categorical variance in the middle, so V600E. I love the fact is that this gets used all the time because I actually used to work in the Cancer Genome Project over at Sanger, and that was one of our variants. Unfortunately, it was before I got there, so I can't claim anything about saying I was involved in that discovery. But effectively, how can you basically go from those assayed variants to that genomic knowledge that is found on the right-hand side and basically classify that information? That is currently going through a vote at the moment to whether the outcomes of that study group, I think it's the outcomes of the study group are good. I believe they kind of are. And effectively, basically, to move into a working group to actually start to develop and test a set of schemas around this. So at Connect, we're going to be hopefully presenting an early alpha version of that model and documentation as well. Sequence annotation, this is now mostly concentrated around the ideas on conceptualizing annotation from multiple different providers. Evan, who leads this, is basically trying his best to conceptualize everything that happens between the major annotation providers, so RefSeq and Ensembl there, but then also actually some of the primary annotation providers, which exist still in GenBank ENA and DDBJ as part of the INSDC collaboration. There are still many people that submit annotation as part of those models still. But this is an ongoing, this is actually a publication. If you have any kind of interest in sequence annotation or this kind of thing, please drop a line and we'll definitely get you looped into that. And then the VA team as well. VA has gone through quite a few different phases here, but again, it's gone through a real invigoration. I know that I've been on some calls recently with Alex and Larry and Matt and Javi, and I can see that there is momentum starting to grow here again. And VA really is this idea of, okay, so you have a variant, so how on earth do you actually link that to some of the kind of knowledge? I know that there's kind of a little bit here which overlaps with the categorical variation group as well, but the water's going to be fine there. And actually is also, if we look over here as well, that we can see that these implementation profiles are things that you are already involved in. So what about some of the other areas of interest that I kind of went through? I thought, what kind of things would this group be interested in? And hopefully I'm going to pique your interest. So one of those is actually quality control for metrics for whole genome sequencing. Now, this is something that has been highlighted as a requirement by, I can't remember which was the driver project that basically flagged this up, but effectively to say, is there a way that we can actually have a more formal description and definition of these metrics and actually understand what they are and then basically go off and implement pipelines that could actually generate it? So if you at any point you thought that QC is actually quite important, I'm sure we have all thought that. Again, this attempts to standardize some of those different QC metrics that you see appear and to ensure consistency between those as well. GATK are on board, Illumina are on board as well in this. The other one, I'm going to put my hand up here. So RefGet, I am one of the people that basically developed RefGet. The thing that is coming up there, though, is I'm going to point towards the sequence collections, which is about groups of sequences and the associated metadata that surrounds that, which is actually about to go into PRC. But ultimately, this would allow you to unambiguously identify collection sequences such as a genome or a transcriptome or a proteome. And potentially, actually, we think there's applications out to pangenomes as well. But also, we're going to basically try and loop this back into verse 2.0 because we know that there is work to be done over that way. But again, if you've ever had problems between HG 19 and HG38 or anything like that. This attempts to solve that problem. And I'm sure we all use VCF here. So again, future of VCF is concerning how you actually scale VCF to beyond the current capabilities of VCF. Again, if you're interested in ensuring VCF continues to scale as a tool, as a data format, then this is something for yourself. And the last of these areas I'm going to highlight is one called Damask, which is currently going, again, this is going through a study group phase, and it's just gone through standard steering committee voting. And the idea here is it's an idea that's gone around quite a few times, but I'm trying my best to figure out how to conceptualize it correctly and typify it. What can we do to ensure that there is interoperability across our work streams, and how can we ensure that representations of schemas remain consistent across different implementations? There's been examples of verse and adapting verse into FINA packets where FINA packets technically is implemented in protobuf and versus not. And of course, there is always, when translating into different dialects, effectively, there's always going to be some kind of loss going on there as well. So if you're interested in schemas, interoperability, anything like that, Damask might be an area for you to look at. But what I would say as well is that this is currently still a little bit finding its feet still, and it is something that has come up time and time again. So, but I'm hopeful that with Kathy involved, that this is actually going to get a certain degree of traction. So that was workstream efforts for the most part. Some pan groups. I just want to go into what some of those other things that hang around are and other ways of getting involved beyond just the workstream level as well. So one of those is the National Initiatives Forum. This is actually a source of direct contact the Global Alliance has with national initiatives from around the world. So groups such as All of Us, in fact, Genomics England, Australian Genomics, will go into what the actual partners are, but it's trying to bring their voice into Global Alliance and bring their perspective into that, but then also try and feed that back into the work streams. What we're trying to figure out here is how do you encourage the diverse? I think that encouraging diverse input is the key thing here, that we're not led by a subset of national initiatives with a particular outset or mindset, because we know that every single nation is different in terms of how its healthcare systems are set up, how genomics is being integrated into that, but then also the regulatory aspects around that as well, and data privacy issues around that as well. And that goes beyond just saying just GDPR or something like that. So there's a ton of members from Qatar through to India, Finland, Estonia, Seneca. I'm not going to carry on going all the way through this, but effectively there are a lot of members involved in here, and this is a really active group as well, meeting very regularly. So again, if you're wanting that kind of, well, what's the national initiative's view on standards development, this is basically where you could go to. The communities of interest. Now, obviously, one of these you should probably be aware of, and that's the Cancer Community of Interest. But again, this is trying to go a little bit more cross-cutting across our work streams and our driver projects in order to try and find individuals that have got a focus that goes beyond, say, projects like VICC, effectively. Even though there was a tight alignment, obviously, between the cancer world and the Cancer Community of Interest and VICC, it is just trying to elucidate some of those other individuals out there that might be interested. Now, there are three other areas to talk about here. One in particular, I'm going to concentrate on the middle one. So the Global Alliance Implementation Forum is another one of these forums. Global Alliance loves forums, it loves initiatives, it loves implementation. that is attempting to find projects that try to address issues of interoperability by going end to end on those implementations. Perfect example of this is actually the project I mentioned at the beginning, which was the one supporting the Beyond 1 Million Genomes project. As part of that, and with that being an EU-wide project, that is attempting to find globalized implementations, bring them together under one stack to actually say, well, can you basically provide access to genomes from national initiatives and European-wide sequencing experiments through global alliance standards? And the answer is, for the most part, yes, you can, but there are definitely challenges about that. Technical Alignment Subcommittee, that's going to be our next one that we'll go to. And the right-hand side is something that's actually kind of dying away a little bit here. So this is the idea of starter kit as well, which was originally tasked with providing reference implementations of global alliance standards, which was developed by our in-house tech team. But this is hopefully going to be superseded by a recent call for comment, which is called Global Alliance Implementations. But again, we'll talk about that later. So task, what is task? This is effectively issues that go beyond work streams, where there are issues abound concerning harmonization, needs to review beyond kind of the auspice of a single product or a single standard. And the idea is that this group basically produces guideline documents and submits them out for approval, and then that comes back in again. Task has had quite a problematic time over the past few years through being just from getting sufficient levels of engagement. Got my dog's hope she's okay, hopefully. But it is starting to gain traction through some very dedicated work there. One of the things that has actually recently gone out is this was effectively a canary down the mine to try to reinvigorate task, was about coming up with a formal procedure around how we would actually write up some of the things in there. So this was specifically about pagination. My gambit here was saying if we can't agree on pagination, then we can't agree on anything really, or at least we can't agree on something that is not as simple as pagination, but at least on what we're trying to achieve. And pagination was a very good idea. This is actually something that was tasked to us years ago, and actually something that was brought up again at our last year, our April Connect meeting, which was in London. And still there was a desire around to see this. So this review period has actually closed, and next week we have our task meeting to actually review the feedback around that as well. So what other issues are there? One that might be interesting in is citing global alliance standards, which has morphed into proposals around what do we need to do to generate DOIs via Crossref. So this is actually a project that the tech team that I work with a lot is undertaking at the moment, and we have mechanisms for actually generating DOIs and starting to submit those to Crossref. But really, we are looking towards task as being able to tell us the kinds of things we should be assigning DOIs to. But that's quite a pertinent thing, really, I feel, and quite useful for Global Alliance. The other side here is there's the media types and issue here about registering those media types, one around locating objects of interest, and the final one about moving crypt GA4GH into its own repo. That's about spinning out, but actually you can start to see that what happens as a little seed of I want to do something actually can quite easily become, well, all right, well, what is actually the procedure for doing this? If you want to get involved, they happen pretty consistently and quite often. I will change that by the next steering committee meeting, because I'm pretty sure that no one actually cares about the steering committee unless you're a part of it. But ultimately, there are documents that appear periodically. There is a new RFC format that we're trialing at the moment to see whether this actually helps with us actually sending out issues to people and to describe them. But also, if you have issues as well that you feel go beyond just a single work stream or a driver project, that's something that can get flagged there as well. And of course, joining up with Slack or the GitHub spaces is good. So the final bit is our process for culture review. Our process has changed very recently, within, say, the past, I'd say, 6 months or so. Normally, everything that goes through Global Alliance now has this open review period, which normally lasts about 30 days, but it can last for longer, depending on how much input has come in. And these basically happen for pretty much everything. That goes from things that get voted out at the Standard Steering Committee through to things that come out from task, and also just proposals from staff, from exec, actually from anyone, I would say, to be frankly honest. But this is a nice way of basically, if you have an idea, there is a mechanism available to us to actually send that out to interested parties to actually comment on. There's 3 that are currently active at the moment, one of which is this Global Alliance Implementations Criterion Catalog, which the aim of that is trying to typify implementations that could serve as reference implementations of our standards, with a way of basically breaking the idea that that has to be developed either by Global Alliance or by a subset of individuals. And actually that spins out a little bit further as well about formal recognition of when, say, things are not, perhaps they're closed source, but they actually do implement the standards. Another one is the is the leadership committee, senior leadership committee membership nominations. And the last one that I did put in there is the task pagination feedback meeting, which is also, it's an open invite as well for anyone that wants to join up with that. So how to get deeper into Global Alliance? Obviously things like meetings like this one, and other GKS meetings as well. If you're not in the Slack space, please join up with that. I think that's actually probably the easiest way of keeping in touch with Global Alliance, but actually still kind of to actively stay in touch with it. There's tons of calls for feedbacks and reviews, and then there's also these other committees that basically you're project members. So your input is extremely valuable to Global Alliance and ensuring that the standards are actually fit for purpose. And with that, I believe that will be it. So I'm just going to stop sharing there. I can see that there's 2 chats hanging around in there. Oh, good. Yeah, Beatrice is definitely the best. And yes, and please, if you want a first point of contact, please contact Beatrice. She is going to make sure that you get to the place that you need to get to.
Thanks, Andy. That was a great overview of, kind of the internals and goings on in the GA4GH. I'd like to ask the group if there are any questions while people are thinking about them. Perhaps I'll lead with one, which is, there are a number of different places to get involved, and a number of different standards that are ongoing. And sometimes there are data interoperability challenges faced by the people of this community, where there might not be an existing group that's focused on that problem domain. What's the best way to advance new ideas and work towards new standards that might help with your individual program interests?
That's a brilliant question, that is, because that comes up all the time. And actually, the other challenge is sometimes where we actually have an overfit to too many of the work streams. So effectively, whatever happens in terms of new development, we always like to co-locate that with at least one of the work streams, because at least then there is a mechanism that keeps the individual in touch with some form of leadership within Global Alliance. It also gives them the secretariat support that comes from people like Beatrice as well. But actually, the first place to go with that is someone like Beatrice, to actually have a proposal written up to say the problem area, like scope out the problem question, send that to Beatrice, and it will basically feed back up to, at the moment, I would be informed on that, but then so would Angela and Peter and people like that. And then there'd be discussions and trying to find where the right place to actually locate this is, and discussions with the work streams. I don't think we found anything yet that doesn't fit tangentially, at least with one of the work streams. I think we have good coverage there, but I'm always up for a challenge around that. So yes, Beatrice is definitely your first place to go into that.
Other questions for Andy? All right, you've done an excellent job of
Excellent.
conveying the landscape to the group. There's a follow-up presentation. So basically, I wanted Andy here to help orient us to GA4GH, but I think it's also important for people to see how the work being done in the VIC is helping to advance standards development in GA4GH. I've got a brief showcase of a few different community projects that are ongoing from VIC. And how they're helping us with our goals of standardized variant interpretation. So I'll go ahead and share my screen. And I will try to get this to show up in slideshow. So I'll ask what Andy asked. Can everybody see this all right in slideshow view? Great. Thank you. Okay, so basically, this presentation is about how we've been advancing work in the Genomic Knowledge Standards workstream. But before we really dive into it, I want to point out that there are a number of other activities the VIC is involved in, or VIC members are involved in different work streams throughout the GA4GH. But since the bulk of our engagement is with GKS, we're focusing on that area today. So I think for those of you that have been around for a while, you know that the VIC started as a GA4GH driver project, basically kind of nucleating at GA4GH. And it was at an AACR meeting of GA4GH associates where our driver project was developed. And we were renewed as a driver project in 2019 during following the reorg, and then again in 2023, starting in 2024. We're an open consortium. We have several hundred members across 25 countries. Our goal is really how we standardize curating, representing, interpreting clinically relevant evidence for genomic variants in cancers. And you are all aware of the types of things that we're trying to model here. These are statements about the role of fusions in driving therapeutic resistance, or point mutations that are diagnostic or prognostic for a particular tumor type. And our goal here is really how we can help computers understand it. And in the Genomic Knowledge Standards workstream, as Andy pointed out, there are kind of like three primary developing products: variation representation, variant annotation, and sequence annotation. The first of these, VR, we'll talk a bit about. So early on, we developed this, the VIC developed this harmonized meta-knowledge base. This is an early one of our works where we basically looked across 6 different community knowledge bases and tried to harmonize the content within them and look at the gaps and challenges in doing so. And one of the key findings we had was that variants that existed in that harmonization effort were not really found across all the knowledge bases. And in fact, most of the time, they were found in only a single knowledge base, which are indicated by these light or these pale yellow circles here. And it's not to get way out into the tail here that you see variants that appear. So here are 8 that appear across all 6 knowledge bases. So as you can see, this very long tail kind of speaks to this challenge of, okay, variants are disparately curated across these resources, which means knowledge sharing is good, because then we can learn about more variants by having interoperability between these. And so one of the key takeaways there was that over 3/4 of the variants that were assessed were unique to just one of the 6 knowledge bases, and these are the 8 that were shown to be universal. And it's not just variants, of course. There's a lot of even 60% of genes were distinct between these knowledge bases, and even more publications. So there's quite a diversity of literature and knowledge that exists across them. Again, emphasizing this idea that data sharing is going to help us do a better job of interpreting. And indeed, one of the things we looked at was, if we aggregate content across these knowledge bases, how good of a job can we do in interpreting? And when we compared it to the Project Genie cohort, we found that there's a big increase in terms of the amount of patients that could be interpreted by virtue of integrating across these. But it could be a much larger amount if we're able to also include evidence that we really couldn't model at the time. And so an example of this is, is this genomic variant, a a G13S mutation, G13C at the G13 codon in exon 2, et cetera. These are all different flavors of categorical variants to which knowledge is associated that kind of require human matching right now. And the idea here is how can we move that to computational matching so that the variant scientists are focused more on review and integration of evidence and less on initial assessment and search for evidence. So to accomplish this, the VICC's been participating in development of the variation representation specification. As Andy mentioned, this is used in a number of other standards across GA4GH, including Beacon and Phenopackets as the variant model. I'm not going to go into too much detail about it, but it is useful for semantic precision of variation that can be used across different types of evidence and knowledge statements. And it also allows us to do neat things like create globally unique computed identifiers, where you can have the same variant that exists across, for example, 3 different reference sequences that ultimately results to the same computed identifier without any sort of coordination between these resources by virtue of being derived from the underlying data itself. And VERSE also helps us with a lot of the challenges that we face in cancer in particular, including things like how we represent copy numbers or copy number changes like low or high amplification or partial or complete loss. So you can find out more about our related work on gene fusions at fusions.cancervariants.org. And this is one of those areas where, we're really helping to drive what's coming up next. So, this was a community effort between VICC, the CGC, ClinGen Somatic, the Cytogenetics Committee from CAP and ACMG, AMP, and others. And we, worked with Hugo and the ISCN and the HGNC and the HGVS committees to basically align our recommendations from this work. And now, of course, the VICC fusion model is part of the HGVS recommendations for a nomenclature for gene fusions. And so this is the release information from the HGVS version 21 release. And what's nice about this is we're now connecting this notion of nomenclature directly into an information model that exists in the fusion specification. And this fusion model is being used to drive the development of the GA4GH verse SV work, which is upcoming. And so this is a work being led by Daniel Cameron of Illumina and Reynold Thomas from EPIC. They're working on developing this verse SV adjacency model. An initial schema is already part of our verse 2.0 repository. And what we're working on now is just preparing kind of a complete release for introduction and discussion at GA4GH Connect in a couple of months. This is an open meeting. People are encouraged to attend virtually, if not in person. If you're interested in being part of that and you don't have the information, please do contact us and we will be happy to get you the registration details. Also, of course, there's the categorical variation work Andy mentioned briefly. This is the CatVerse, and you can we'll be talking a bit more about it at Connect. But in short, it does help us address some of the complexities of how we represent what we call variants or categorical variants in many of these knowledge bases that we have in the VICC. And this includes a whole slew of different ways of thinking about variation based off of function and quantity. Reading frame, where it is in the where it's located in the genome, et cetera, and across multiple different levels, all the way from the chromosome up through kind of a systemic tissue level. And this work's being led by Daniel Puthawala. And this basically helps us address this issue of, okay, we have a genomic variant. What are all the different ways that you might think about that variant as a change of function, something in the BRAF gene, it's in frame, a change of sequence, et cetera, that might be associated with knowledge. The other area that we're focused on developing within GKS is the variant annotation work. Our colleagues over at the ASH Somatic Precision Medicine Group have developed this variant database for common genes and variants associated with hematologic malignancies. They gave an abstract on it in 2020, and one of the key findings from it is basically that there's a lot of underlying evidence where we just kind of know evidence was assessed using these different resources But whether or not, that was unique or the same, or, if it was differing from those used by other institutions is not clear. And so that's what all these red boxes are for. And then, of course, some of them also had like in-house stuff that was unique to their own assessment, as well as some things that were used only by that group, but are public-facing tools, so could be used by others. Those are the yellow boxes. And what's interesting is that, like, across these, we begin to see this pattern of, okay, there's a lot of reused resources, but the underlying evidence driving these interpretations is unknown. And that can create challenges in terms of, well, how do you integrate the classifications made by 6 different institutions? And in fact, that was one of the limitations of this work. And so in order to get around that, we need standards to help us integrate evidence and knowledge, and therefore, quite specifically represent it. And so there's this joint work going on in the GKS workstream to pilot the application of the GA4GH genomic knowledge standards to some of our favorite clinical variant interpretation resources. Now, this includes CIVIC and ClinVar listed here, but in our initial work, we also included the Molecular Oncology Almanac, which I think we showed in a couple slides. But the idea here is that these 2 driver projects are really working to aggregate, standardize, and integrate knowledge across variant curation platforms. And we're doing this using the VA spec, which is a draft specification that's being led by Matt Brush and Javi Lopez. This specification is focused on this core statement model shown here. I'm not going to go into detail just for time constraints, but in general, the idea here is that the VA model allows us to associate evidence with evidence lines like codes to higher order assertions. And so we implemented this, as I said, for CIVIC, the Molecular Oncology Almanac, and ClinGen did this for the ClinVar database. And we created a snapshot release for each of the last 2 plenaries for GA4GH. And this work you can find at GA4GH/GKPilot. Some of the more recent stuff that's going on to help drive our oncogenicity framework is the development and application of these standards for resources used in the VICC, ClinGen, CGC oncogenicity guidelines. And this includes the use of nomad population frequency evidence. So what happened is the nomad team basically applied the open source verse tooling in order to annotate the 1.9 billion alleles of the V4 data release with the verse information model and identifiers. And these are all available inside the nomad Hill tables. In addition, a variant annotation specification draft model for cohort allele frequency was created. So that allows us to basically create these sorts of low-level objects about allele frequency and associate that with the higher order statements of BA1, for example, for ACMG AMP, to drive higher order statements of its benign classification. The data model itself looks like this. We won't get too into it, but in short, all this stuff in blue, these are kind of the key attributes. And then we also have other measures that are associated with it, including the notion of depth and quality filters, and as well as ancillary results such as the group max FAF, which is one of the key things that's hosted by the Nomad resource specifically. And then finally, VERSE is helping us in terms of translating functional data for use with the oncogenicity framework. It allows us to provide contextual information about homology maps from the MAVE experimental sequences that are used to the corresponding sequences in human reference assemblies like RefSeq. And so this work, preprint went out mid last year. It's still under review at Genome Biology, and I guess there'll be more coming out about that hopefully soon. But this work's being led by Jeremy Arbesfeld in partnership with our colleagues over at the Atlas for Variant Effects Alliance. All right, so I did manage to get through what was supposed to be a 30-minute presentation in about 15 minutes. So I know that was a whirlwind. I'm happy to answer questions. But before we get there, I just want to say one more thing, which is if you're interested in participating or leading any of the GA4GH activities as a VICC Driver Project representative, let us know. This is a great way to get involved in the community, engage with other works that are going on in the, across the VIC, and ultimately to help consolidate and synergize on our efforts. So if you have an interest in doing so, please reach out. My email address is here. And with that, just a few acknowledgements. These are only the leads of the various teams that worked on all the stuff that we covered here. As you can see, it's a very diverse set of people, and there are dozens, if not hundreds, of additional people that are not shown on this slide just by virtue of not having the space. So big thanks to everybody. And with that, let's discuss.
I think that was a really nice presentation, Alex. And also thanks to Andy for his presentation before. It gives a really nice kind of like 10,000-foot view of what we're doing, where we're going. Yeah, it was really good to kind of take a step back and look at the big picture. So thank you for that. If people want to join the GA4GH Connect that you have mentioned a couple times, is there a link for that? Is there something that you could share with this group?
Yeah, absolutely. I think that I'll reach out to Beatrice and get the appropriate announcement, as well as the appropriate registration link, and then I can send it around to the community along with the recording from this call.
Awesome, thanks.
All right. Well, if there are no other questions, thank you all for your time and attention today. Wes, do you happen to know who's coming up at our next call?
Yep. Our next call is going to be Kathy Cottrell and Karen Sushaya are coming in and giving an update on the gene fusion oncogenicity specification and how that's going.
Great, fantastic. So this is one of our, it's our second newest project, with our newest project, of course, being our complex variants group with AMIA. But it'll be great to hear progress from the gene fusion oncogenicity folks in the next call. So thank you, Wes. I hope you can all join us for that. And until then, everyone stay well, stay safe. See you next time.